Okay, good morning, everyone. Welcome again. This is the last lecture, the second of the mononuclear phagocyte ontogeny. We're going to focus on dendritic cells today. I do want to go back a bit because I thought that there was some confusion in this critical paper and its implications, and you'll see how important it is even in the dendritic cell ontogeny. If you remember, this was the GNU 2010 paper that was the tipping point in changing the previous model, which was the Van Firth model, to one where many, if not most, of macrophages in the tissues of the body are embryonic. In this one, or embryo derived, this one with the microglia especially, or yolk sac derived, in which they used Runx1 Cree mice. Cree ER, it was a Mur-I Cree Mur system to pulse the pregnant dams at E7.25, and then allowed the pups to be born, at which point all the way out to 6 weeks or 8 weeks, and it's been done even later, you can then identify YFP microglia originating from that TAM pulse, and essentially has led to this model 2, which is the focus of most of the literature now, whereby excuse me, at E7.5 you have early EMP, erythromyeloid progenitors, that become yolk sac macrophages, leave the yolk sac, migrate into the brain, the developing brain. And at that point, you have the blood-brain barrier. So no new monocytes are really coming in. And so these macrophages are then left to their own devices to maintain themselves in the tissue throughout the lifetime of the mouse. And there's a lot of evidence in humans showing that this is the case. By contrast, you have these late MyB positive EMPs that come from the hemogenic islands. They also can become, or do become, yolk sac macrophages, but they eventually fizzle out. Instead, you have EMPs that then seed the fetal liver. And these fetal liver EMPs now become fetal liver monocytes. And as you know, the fetal liver is the intermediate place where hematopoiesis continues in the developing embryo. The stage after that is the AGM. And then after that, eventually you have the bone marrow established. But going back, you now have these fetal liver monocytes, and these guys then seed all the tissues of the body in the embryo, or the organs, and they become macrophages. And those guys are maintained without monocytic input for the remainder of the lifetime of the animal, with some exceptions. As I mentioned, there was the open niche in the gut and in the dermis, and there was slow and fast in heart and pancreas. So let me take a moment to see if there's any questions on this model. Not one question about how this works?
So what becomes of the blue monocytes that become blue macrophages in the tissue? Do they again become outcompeted?
So yeah, I think you're talking specifically in model 2.
Yes.
That's a good question. And so this is sort of, again, outside of the canonical thinking at this point. But if you look back at the GNU paper, when they pulsed with Runx1 Cree mice after a certain time point, it seemed to give rise to some hematopoietic stem cell that tagged monocytes for the adult mouse. So there is some contribution there that they can't deny, but it seems to be the minority of those cells. Thanks for the question, Daniel. Okay. Any other questions? If not, I'll move on to dendritic cells. So from that picture, you can see that this emerges here. This is in 2015. Remember, that article was in 2010. Obviously took some time for consensus to be built. But this is essentially the picture now, that you have your yolk sac derived macrophages, which then seed mostly the brain, to some extent the dermis, and then you have late EMPs that seed the liver, that become fetal liver monocytes, which then seed the tissues and become macrophages. Okay, and these are distinct from now what we'll review are the dendritic cells and monocyte derived cells, which are all adult bone marrow HSC derived. So on the left-hand side, we have the CDP, and I'll talk about this, the common DC progenitor, and the pre-DCs that become DC1 and DC2. Here's another look at this algorithm. Again, on the left-hand side, you have the embryonic origin macrophages, and this is distinct from the adult HSC origin, which give rise to the monocyte lineage and the dendritic cell lineage. So what we're going to focus on today is the dendritic cell lineage, with particular focus on the classical or conventional dendritic cells, not necessarily the PDCs, the plasmacytoid dendritic cells. There's still lots unknown about plasmacytoid dendritic cells. They're not canonical, conventional, classical, if you will, in their antigen presentation to T cells. So I prefer to focus really on this conventional subset here. And it was sort of, the nomenclature here was established in this article, 2014. Again, note that this is 4 years after the Genu paper. And what they had shown, and I'll show you what they had established, or what they had proposed rather, was that there were essentially two buckets or two bins in terms of the DC phenotype and function. There's DC1, CDC1, classical or conventional, dendritic cell type 1, and CDC type 2, which had two different transcription factor dependencies and had functional distinctions as well. The DC1, as you'll see, or I'll talk about, triggers more of a T helper type 1 response. It's cytotoxic T cell response. Whereas the DC2 is more specific to type 2 or type 3 responses, type 2 being CD4 T helper cells, type 3 being TH17, IL-17 secreting T helper cells. So they have both distinctions there. Now, as I like to do, go back in history. The first report of dendritic cells was by Ralph Steinman and Zanvil Cohen. This is in 1973. What they did was to basically isolate a population of cells in the spleen that looked nothing like, well, I shouldn't say nothing like, but looked distinctive enough from macrophages to consider it its own population of cells. These were veiled cells. They were highly arborized as compared to the macrophages. And they were really, really, really good at stimulating T cells in a dish. And so they thought these two characterizations were sufficient enough to consider it as its own cell. And certainly you've heard of the term dendritic cell, so certainly there was traction there. And to add to that, Ralph Steinman received the Nobel Prize shortly after his unfortunate death in 2011 in physiology or medicine. So clearly dendritic cells have consensus in terms of them being a separate population. But having said that, and you can see that this was published in 2010. Again, I think time, the year is important for context here, that that there was still some confusion. If you look here, if you read just this abstract, and by the way, this is an excellent read. It's interviewing these 5 or so individuals who were experts. If you read here, when Ralph Steinman's Anvil Cohen first described dendritic cells in 1973, it took many years to convince the immunology community that these cells were truly distinct from macrophages. Almost 4 decades later, the DC is regarded as a key initiator of adaptive immune responses. However, distinguishing DCs from macrophages still leads to confusion and debate in the field. Again, remember that this is in 2010. What was the source of the confusion? The source of the confusion is how, at the time, were we distinguishing dendritic cells? Was it by morphology, these stellate processes? The problem with that is there were cells in the body that were part of the MPS system, the mononuclear phagocyte systems, that had the stellate morphology, and even non-myeloid cells, for example, lymphocytes. So gamma delta T cells in the epidermis have a stellate process. And this is more reflective of the tissue itself, not necessarily of the shape of the cell. In other words, the stellate processes are usually there because they're interdigitating between tightly packed cells. How about function? Well, macrophage can certainly stimulate a T cell. Maybe it might not be able to prime a naive T cell, but you can argue that in some cases, and it has been shown that monocyte-derived dendritic cells can indeed stimulate or prime a naive T cell. So the problem still stands there. What about phenotype? For a long time, the CD11C marker was considered to be a gold standard of dendritic cells, but David Hume had pointed out in an article which was titled "The Dendritic Cell Myth," and this was around the same time, had noted that there are plenty of macrophage populations that express CD11C, namely the alveolar macrophages, for example, and the red pulp macrophages in the spleen. So that didn't cut it either. And at the time, Lineage wasn't cutting it as well because we didn't have this 2010 paper really published or even had full consensus at the time. So you can see where the source of the confusion was. So are there any questions on that part before I move on? Okay. So be that as it may, the field continued to go on, of course, as noted by the fact that Ralph Steinman won the Nobel Prize in 2011. This is a depiction of what was of the thinking in 2010 from Liu and Nussensweig. Nussensweig, of course, was the mentee of Ralph Steinman, who established his own lab and now is a very famous immunologist. And just to show you that these the specification in DC poiesis, how this sort of came about, without getting into the nitty-gritty. But there's this paper in 2006 in Science by Frederick Geismann's lab, who basically identified a clonogenic progenitor cell that was restricted to the monocyte macrophage or DC lineages. So once this cell was identified, you can then pulse or you can then feed these cells with FLT3 ligand. This is a ligand for the FLT3 receptor, which is tyrosine kinase receptor involved in DC poiesis. And this would then give rise to the dendritic cell lineage. Whereas if you gave it MCSF, this is CSF1 linking or binding to CSF1R, would then give rise to a monocyte. And I would ignore TIP DC but basically would give rise to the monocyte and the monocyte-derived macrophage. And then shortly thereafter, this came out in Nature Immunology from Ken Shortman's lab, showing that they were able to identify this CDP, which was a common DC progenitor. So now this was restricted to the DC lineage. And then again, after that, or around the same time, there was a paper by Nussensweig's lab showing or identifying what they referred to as the precursor DC or pre-DC. It emerged in the bone marrow where it was committed to a dendritic cell fate, and then was released into the blood where it would take up its position in the periphery and then become fully mature into a dendritic cell. And so this came about in 2009, and again, this is a 2010 sort of depiction of what was thought or what was considered to be the case for DC poiesis. Now, fast forward 5 years later, a number of anomalies has grown. So it wasn't even only that the fact that there was a confusion about what was a macrophage and what was a dendritic cell. Even in the dendritic cell field, there was difficulties in nailing down the exact commitment pathways and fates of specification and poiesis. This is an article by Caetano Reyes Souza, who casts his doubt on the previous model based on lineage tracing work that his group had developed that was anchored on DNGR1, which is a C-type lectin expressed by dendritic cells. And through this, he then showed data to sort of cast doubt on the true existence of an MDP, a macrophage, monocyte macrophage DC progenitor that was committed to one or the other. And he also cast doubt on the common or the CDP for the subsequent step. And so you can see clearly that there was some disagreements in the field. Let's see here, the existence of MDP or common precursor for CDCs and PDCs is debatable. However, CDC restricted progenitors here, CDCP, can be distinguished from PDC, which is fine, by expression of CD115 and DNGR1. CD115 is CSF1R. CDCPs further differentiate into pre-DCs, which acquire 11C and ZBTB46 expression and travel through the blood to peripheral organs. Their differentiation in the CD8 alpha, CD103, and CD11B DCs. This is the DC1 and DC2, respectively. In target tissues is controlled by transcription factor, BATF3, and ERF4. I'll talk about that in a second. And this goes into PDCs, which we'll skip. Monocytes also develop in the bone marrow and arise from common monocyte progenitors, CMOP, distinguished from DCP and pre-DC because they lack DNGR1. So at least there is consensus that there's a CMOP, and these are distinct from pre-DCs. Any questions from this part? So despite the disagreements and anomalies, we were moving towards this consensus about a DC1 and a DC2. That, in other words, there was specification of two DC types, and a lot of the time, for this area, there was a lot of wonder about how T cell subsets emerged in terms of differentiation of Th1 and Th17. Was that determined by the T cell or the microenvironment in which the T cell was primed? Or did the dendritic cell have something to do with driving those different programs? And there was always a sense that there was, especially with the identification of IL-12, moving to a Th1 type of response. But this actually helped to explain how this may arise, at least part of it, because the DC1 was able to trigger was more sort of directed at activating a Th1 response, where DC2s were more activating more of a Th2 and Th17 response. So there were some signals passed on by the dendritic cell, and this helps explain it by the classification of DC1 and DC2. DC1, of course, expressed CD8 alpha in the lymphoid tissues, whereas in peripheral tissues, and I'll show you this in a sec, they expressed instead CD103. The DC2, this is kind of a hodgepodge, and you'll see this. They express CD11B. They don't seem to express CD8 alpha. In fact, in lymphoid tissues such as the spleen, they even express CD4, distinguishing them from CD8. So just to go back in time, this is work from Ken Shortman in Science, and they showed that there was in fact, the transcription factor BATF3 was responsible for the differentiation or specification of these DC1s, and they were able to use this for the first time in vivo when they knocked out the BATF3. And you can see here, this was enriched for CD11C splenocytes, here being the CD8 and CD4, as I had mentioned before. In the wild type, you can see both populations of dendritic cells, the CD4 and the CD8. Whereas in the knockout, you lose this population of CD8 DCs or DC1s. And in the absence of that, they showed that there was an impaired ability to trigger a Th1 response. And I think they showed this in the context of West Nile virus. Now, this was in the spleen. This was work done in the spleen. And then work before that, Ken Shortman, a lot of the people, others who were critical in this field, used the spleen as opposed to the lymph nodes. And the spleen certainly was a lot more simple, because if you compare it to the lymph nodes, in the lymph nodes, you not only had these lymphoid dendritic cells that live in the tissue, but you also had migratory DCs that came from the draining lymph nodes. So the spleen didn't drain from the tissue. The spleen didn't necessarily drain tissues. The spleen filters the blood, whereas the lymph node drains the tissues. And you have DCs that migrate from those peripheral tissues into the lymph node. So you then have this whole gamish, both of migratory and non-migratory DCs, which is not the case necessarily in the spleen. So it seemed like a very nice place to study. And of course, you had the historical implications from Ralph Steinman's group, who initially identified the dendritic cell in the spleen. So that gave the spleen a lot of traction, but had left the lymph node and other peripheral tissues sort of ignored for a long time. And this sort of changed in the mid-2000s. This is a key paper from Miriam Murad's group, who had showed in all of these non-lymphoid peripheral tissues, one could find two main types of dendritic cells, one that had a CD11B expression and the other which had the CD103 expression. So you can see that in the islets, in the liver, in the lung, and in the dermis. And you can also see it in the intestinal tract, in the lamina propria, but there was a third population there, and they had a separate paper for that. But it pretty much fell in these lines. And if you looked at the CD103 versus the CD11B for these markers, you can see that the CD11B had higher expression of CX3CR1, SERP alpha, F480, whereas the DC103 had expression of Langren, for example, and then were lower for CX3CR1, SERP alpha, and F480. So there was a similar sort of program or expression profile for these DCs, in all of the different tissues, signifying that they may be in fact derived from a single lineage. And this is just to show you some more data from the paper. Again, looking at 11B versus CD103, what they did here was to knock out FLT3. And when you knocked out FLT3, you essentially lost the CD103 population. And so that was really nice data indicating that these not only were dendritic cells, but there seemed to be a lineage of these dendritic cells, quite important, or quite requiring FLT3. On the other side, you have CSF1. Now this is tricky because they would knock out the CSF1 receptor in this experiment, and what one found, or what they found, is that there was a sort of a loss of CD11B DCs, whereas there was a gain of CD103. So that it made sense in that there are more CD11, more CD103, because if you lose one population, you gain the other in terms of percentages, right? But the major point here is, in the absence of CSF1, you had a reduction in these CD11B cells. I guess this was not the case in the liver. So what this suggested, again, was that there's really two populations of dendritic cells. So there's a DC specification. There's the DC1, which wasn't called DC1 at the time. And these were CD8 or CD103 DCs. And now you had the second population, which were CD11B, which were 103 negative. And what Ken Murphy's group did here in 2010 was essentially show that what was known in the lung, what was known in the spleen with respect to this type 1 or CDC1 dendritic cell triggering a Th1 response was the case also in non-lymphoid tissue. So now he's unifying this idea that what they found in the spleen was consistent with what was found in peripheral tissues. So that CD8 DC is equivalent to the CD103 BATF3 dependent DC in peripheral tissue, namely here, which he's showing in the lung. So for example, here in the BATF3 sufficient mouse, you have both populations. This is showing you the CD103. This is showing you the CD11B. And then when you use mice with a knockout BATF3, you can see that this population is no longer there. And that's basically shown here in the analysis. Here, what they're showing is FLT3 versus CSF1R. If you remember, there was that distinction shown by the Murad group. CD103DC expressed more FLT3 receptor, whereas CD11B expressed CSF1R. Now, the problem with the CSF1R caused confusion. So it seemed to be clear. It seemed to be clear that the DC1s, okay, the CD8 DCs seemed to just sort of easily fall into this group or bin, okay? So they either express CD8 or CD103. You can they're BATF3 dependent, okay, and if you don't have BATF3, they disappeared. They didn't show up in the tissues, and so there was a number of categorizations or number of characterizations that made sense and continued to show up in these experiments, whereas the DC2 or the CD11B was a bit trickier. One of the problems was that it required CSF1R, and that distinguished it from what was known about dendritic cells versus the monocyte-derived cells, in that monocyte-derived cells required CSF1R. Microglia and other macrophages required CSF1R. And so if these were truly different cells, why are they both relying on CSF1R? And that still kind of remains to be a problem in the field. Now, one important paper by Ginou's group have sort of helped resolve this issue to some extent, which was to find out that the CD11B DC in the periphery seemed to consist also of a monocyte-derived cell as well. So let's look at this. They also showed that it was ERF4 dependent. And so let's just go through the data here. This is in the lung. They've identified your dendritic cells, which are MHC class 2 high, CD11C positive. And now within this population here, you have a CD103 and a CD11B. So now within the CD11B gate, what he found, or what the group found, I should say, was that there were two populations. One was CD24 positive, and the other one was CD24 negative, CD64 positive. And they had shown that this CD64 was partly, this population was a contaminant of the DC population. And these were in fact macrophages, monocyte-derived macrophages. CD64 is the FC gamma receptor 1. So when they now sort of pulled that out and looked at these comparatively, things started to make a little bit more sense. If you look at, let's say, so you have here is the expression levels of each one of these populations. CD11C makes no real difference. Of course, CD24 is expressed on both DC populations, but not the macrophage population. CD26, BTLA. BTLA is a DC marker, and you can see that fits nicely because the macrophage expresses low levels. GR1 at the time was used as a monocyte and neutrophil marker. So clearly these are not monocytes or neutrophils, even though this macrophage may be monocyte derived. MHC class 2, you can see that both DC populations express it. The macrophage also expresses high levels of MHC class 2, so that didn't help. F480 is a macrophage marker. And so the expression of F480 here on the macrophage made sense, but you can see it's also expressed on these CD11B DCs, which is okay. And you go so on and so forth. Another marker here, which really made sense in this distinction of CD14, this is highly expressed on monocyte macrophages. And that makes sense with this designation here with CD64. And here you can see now they looked at the lung draining lymph nodes. So remember, the lymph nodes contain both populations that are non-migratory and migratory. They were looking specifically at the migratory population. So when you're doing flow cytometry of draining lymph nodes with the CD11C and MHC class 2, you see two populations here. This population here, which is not gated on, is the non-migratory population. These are DCs in the lymph node, which have not migrated from the periphery, they live there. This population of MHC class 2 are considered migratory. And if you look within this population of migratory, you can now see CD103 and CD11B DCs. And if you look within the CD11B fraction, you only find the CD24 positive. You don't see the CD64, which makes sense in the sense that CD64, these CD64 max are not thought, or macrophages in general are not thought to be able to migrate from the peripheral tissue to the lymph node. That is a key feature of a canonical dendritic cell. Okay, so that made sense. So now focusing on this right side of the graph, what you see here is now they're looking at FLT3 and CSF1R. So in this experiment, you can see that they're looking at the three different types of cells that were pulled from the lung. The CD103 DC is in white. You can see it has very high FLT3 expression. The CD11B DC has lower FLT3, and the MAC has almost none. If you look at the CSF1R comparatively, you find that the CD103 has none. The CD11B has expression of CSF1R, and the macrophage has even higher amounts. So you can see where this confusion is. This DC2 CD11B DC has sort of almost, it really looks like a macrophage in many ways. Even when you look at its markers, for example, F480 is a macrophage marker, but yet it's considered a DC. So it's almost like a hybrid macrophage DC type population. And that's can really kind of maintain some level of confusion in the field. If we wanted, we like to put things in pristine black and white boxes, and this one didn't fit. The CD103 fit pretty well, but the CD11B DC does not. So let's look at these. These are bone marrow reconstitution experiments. Remember that DCs are bone marrow derived, so you can wipe out the bone marrow with irradiation and then give it bone marrow from a knockout and see whether it replenishes and what population it's able to replenish and which population it's not. In this case, you have a wild type. In this case, you have a wild type and you have FLT3 donor bone marrow. And if you give, excuse me, knockout donor bone marrow, you're not able to establish, and they can tell by these congenic markers what's donor. So CD45.2 marks the donor. If you use FLT3 knockout, you're not able to reconstitute the CD103 population, whereas in wild type grafts were. FLT3, you were able to establish some, even with the FLT3 knockout, you were able to establish some DCs, some CD11B DCs, but clearly this is substantially lower. And then what they're showing here in the FLT3 knockout, there was really no problem establishing these monocyte-derived macrophages. Remember, monocyte-derived macrophages are bone marrow derived. They're not embryonic derived, okay? So this comes back again to the problem, really monocytes have been a real tricky player in establishing ontogeny, understanding ontogeny, if you think about it, because they can become one cell type or the other, and they express a lot of the same markers. And so I think it was monocytes have been a red herring for a long time in the ontogeny studies. Now, if we look at the CSF1R knockout situation, of course, you have no problem with the CD103s to reestablish themselves, even though there's a knockout of CSF1R. But what's interesting here is that they're showing that the CSF1R in the knockout here, there doesn't seem to be a problem in establishing the CD11B DCs. So even though they're expressing CSF1R, it seems like these guys are not dependent on CSF1R to establish themselves, whereas the macrophage is. So again, this comes back to this issue of CSF1R being more of a monocyte related phenomenon or related hematopoietin for monocyte-derived macrophages. If we look at the migratory population in the lung, in the wild type situation versus the FLT3 knockout, you can see there's clearly a loss. The CD103 is not able to reestablish itself in the FLT3 knockout, completely consistent with everything. The CD11B DC to some extent is in the FLT3 knockout, meaning that there are some DCs, and sure, this is statistically significant, but this suggests that there are some DCs, CD11B DCs, maybe this is still part of the contaminant, that seem to be able to establish themselves in the absence of FLT3. FLT3 is critical. We know FLT3 is critical for DC poiesis because in the absence of FLT3, you really have a severe loss in the numbers of dendritic cells systemically. And then through traditional in vitro cultures, you really need FLT3 to push, if you remember the MDP and the CDP studies and pre-DC studies, to push it into a DC designation. And we know also from FLT3, provision of FLT3 through FLT3 secreting tumor cells greatly increases conventional DC numbers in mostly in the lymphoid organs. So FLT3 is a really critical piece of DC poiesis. And so you have to ask yourself in the FLT3 knockouts, when you see CD11B DCs such as these able to show up, you have to ask yourself, is this a contaminant potentially? And so let's just sort of summarize this, that CD64 was a really helpful marker, and it still is to this day, in helping decontaminate, I guess it's the word, CD11B DCs from non, let's say monocyte-derived macrophages from that population. But it itself is not absolute. We know now from subsequent studies that there are populations that of DCs, canonical DCs, that express CD64. So again, it's been helpful, but it's not absolute. And it again highlights this problem of the DC2 problem, the CD11B DC problem, is whether it's a heterogeneic bag of cells, or is it truly its own population, and whether it's really purely a dendritic cell or not. Any questions so far? I know this is a tricky, very tricky part, but it does help me to know where you guys are in terms of whether I'm missing something, if there's something that can be explained a little bit better.
I have a quick question. If the FLT3 deficient or knockout CD11B population is not a contaminant, would that imply that there's two distinct populations even within that? I think I missed you saying that. But
Can you say that question again? I'm not sure.
Yeah, if the CD11B dendritic cell population that is arising in this FLT3 knockout condition, if that isn't a contaminant, would that imply that there's even two distinct lineages in that population as well? Or
so that you're hitting the nail on the head.
okay.
That is part of the problem. Is it you're right, is this a heterogeneous bag of cells, right? And
Yeah.
yes, it could suggest that. It could suggest either or. It could suggest that it's either a contaminant, or that there are different potentially different subpopulations within that bin, if that makes sense.
Is there any idea what the other population might rely on if it did exist? If it wasn't just a artifact or a contaminant?
Right. So the answer is no at this point. I could move on to the next slide to address that to some extent, but there's still no real consensus on that point. What we do know is that ERF4, the transcription factor, seems to be really critical for the emergence of DCs. To answer your question about possible subpopulations, there is some evidence that that's the case. You can see that in showing here in this ERF4 high category, there are two different populations. One of them is ERF4 dependent, one of them is less dependent. And then one is really seems to be contingent upon KLF4 versus the other one, Notch2. Again, these are early. These are early days for this sort of analysis, and this is what the picture. This is kind of what the picture that is emerging from these studies are suggesting.
Okay, thank you.
So then, can I just follow up with a kind of tangential question? So
Yeah.
just looking at our not complete understanding of DCs and things like that, just looking back basically to the immune privileged sites, you mentioned that it seems perhaps antigen presentation from cells that are coming from those sites are tolerogenic. Is there any evidence to suggest, at least so far, that the heterogeneity that we see in, for example, whether it be CDC2s or other antigen presenting cells, would indicate that those sort of, I'll call them tolerogenic cells, for lack of a better word, exist, or is it just a completely unexplained phenomenon?
I think what you're asking is, can we explain immune privilege through the existence of one of these dendritic cell types? Is it one of these dendritic cell types that might be prone to immune privilege, to helping
Essentially, yes.
or tolerogenesis?
Essentially, yes.
That's a big question, and it's been very problematic one to answer. There is a substantial literature in the mid and early 2000s that you could generate tolerogenic DCs. At the time, they weren't thinking necessarily of DC1 and DC2. What they were thinking about is the relative expression of MHC2 and co-stimulatory molecules. And the theory was that if you had low MHC class 2 expression, which explains immune privilege, the absence or lack of MHC class 2 expression. So if you had the low expression of MHC, but at the same time high expression of co-stimulatory molecules, that engagement of T cells in that combination would generate a regulatory T cell as opposed to a effector or helper T cell. And to a large extent, those experiments worked, but it was hard to really show that this T regulatory response was very durable, long lasting. And so it may sort of resemble something that's more of a momentary possibility as opposed to a real long-standing, canonical path. I don't know. I hope that helps.
Yeah, I think so. The biggest thing that I'm wondering essentially is, how would a cell be able to, this is more of a rhetorical question, but like, how would a cell coming from one of those sites, I'll say, have the memory that it was at one of those sites, and that the antigen that it's carrying should be tolerogenic to know to be able to do that pathway. So
Okay.
I'm wondering if it's more of like a physical characteristic. So for example, it's its own independent cell population, or if it's some sort of a physiological process that for some reason happens with different antigens. But again, it's, I think, a bit too tangential for this.
Well,
setting.
I like it because it brings together both lectures, and it allows me to highlight a couple things. So I appreciate the question, Cole. So think of it this way: DC poiesis happens in the bone marrow. You get a pre-DC. And by the way, and kind of what I'm showing here is that the pre-DC1 and pre-DC2, there's evidence that that specification begins in the bone marrow. Okay. And then they leave as pre-DCs and they enter all of your tissues, not yours necessarily, but the organism. And once they get into the organism, then the organism influences their function and their maturation. So what enters the lung in a non-immune privileged tissue, right, versus the cornea, which is an immune privileged tissue, essentially should come in as the same input, but the output is different. And the output is different because of the immune privileged nature, what that tissue is exposing that dendritic cell with. So to give you a sort of solid example, in the cornea, the dendritic cells there are bathed in high levels of TGF beta. And so the theory is that, and this has been shown in a dish, that with the capture of antigen in the presence of high TGF beta, you're then reprogramming the dendritic cell to have more of a tolerogenic fate in terms of when it goes down and interacts with the T cell. Okay, so yes, does that make sense, Cole?
Yeah, I think so.
Okay. We only have a minute or so left. Are there any other questions? This is the last lecture, so if you do have a question, this is the time. If not, that's okay too. You can always reach out to me by email. But let me just again end with this slide here. This was the most current sort of opinion piece. This is by Ken Murphy and Genu, and I saw that it was reviewed by Catano. So I think that this is a good representation of where we are in the field right now. You see that there's no they still are including the MDP here, and they're still including the CDP, and I think there's plenty of evidence to keep that there, although there are some confusions or anomalies to that model. And then the other thing I do want to mention again is that this DC1 and DC2 specification begins in the bone marrow. So their imprinting of one fate versus the other begins in the bone marrow. Okay. All right. So without any further ado, thank you. It's been fun. Feel free to email me again, and good luck with the rest of the class. Okay. Thanks, everybody.
Thank you.
Thank you.
Thanks so much.
You're welcome.